But I told you yesterday also, we are left with only two topics in this chapter. First is nomenclature of enzymes. And second, I have just taken up an extra topic for you. And that is some interesting facts about enzymes. Though that has nothing to do with your syllabus, but you will find it interesting. Some interesting facts about enzymes. So let us start the second last topic of the chapter. It is classification and nomenclature. Everybody knows what is classification, but what is nomenclature, beta?
Naming.
Naming of us.
Yes, exactly. The naming of something is called nomenclature. Earlier also you must have come across this word nomenclature. Where? In what?
Chemistry. Alkanes, alkenes, alkynes.
Biological
Okay, that is
Biological nomenclature.
yes, we have biological nomenclature. We have IUPAC nomenclature. Just now one child just told me about chemistry, alkane, alkenes, that we have classification. So that is IUPAC nomenclature. What is full form of IUPAC?
International Union of Pure and Applied Chemistry.
Yes, very good. So we need to keep remembering what we have learned in earlier classes also. ये नहीं कि पीछे का सब भूल जा रहे हैं. That we need not do, beta. Okay. So we are going to do nomenclature of enzymes today. Not the entire biological nomenclature, just the enzymes we are going to study, how the enzymes are named. Let me tell you about the old method first. It was a very simple method. They just said कि root word के साथ A's लगा दो, ASC A's. Root word means keyword, main word. Whichever is the main word, just add the suffix ASE. For example, if your substrate is maltose, so the root word is malt. The root word is malt. So if you want to name the enzyme, simply write maltase. One more. If your substrate is sucrose, then you know that the root word is this SUCR. You need to simply add. Yes, it becomes sucrase. So wherever we had ASE, ASE, ASE as a suffix, that means it is an enzyme. If there is an ASE, then it is an enzyme. This is the oldest method of nomenclature of enzymes. And because beta ये इतने सालों से use होता आ रहा है कि still we keep on using these names. Many of the times maltase, sucrase, we still keep using them. This is one of the oldest system of classifying, naming the enzymes. But बहुत सारे years तक, because we kept on using this system only, so still in many books you will find the naming by this system only. Now let's proceed further. A little advanced system, that is your second method. This is, you name the enzyme on the basis of its source. From where are you getting the enzyme? So name it from the source. For example, here we have, there is an enzyme which is called bromelain. Bromelain. What is the name of the enzyme?
Bromelain.
This enzyme source, it is extracted from pineapple. And we know that the family to which pineapple belongs is Bromelaceae. The family is Bromelaceae. So the source was brom. So we have taken this name from here and it has become bromelain. No need to learn these examples. No need to learn the examples. I'm again telling you. These examples are just there so that you can understand the system of naming them. You don't learn these examples as such. So jaldi se karte hain. Which is the oldest method? What is the suffix that we need to add to the root word?
ASE.
So when I say maltose, then root word ho gaya hamara malt, and we are going to add which suffix if we have to name an enzyme?
ASE.
AC.
Enzyme. Very good. Your enzyme becomes, what is the name of the enzyme?
Maltase.
Similarly, if you talk about the sucrose sugar, then the enzyme would be,
Sucrase.
Sucrase.
Race.
matlab, wherever you have an ASC, you have that is an enzyme. Let's move on to the second method. Second method was based on what? The source
Okay.
of the enzyme. Source means आपको कहाँ से वो enzyme मिल रहा है? Where are you extracting that enzyme from? I just give you an example of an enzyme. What was the name of the enzyme?
Bromelain.
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 Is
Bromelain.
extracted from which fruit?
Pineapple.
Pineapple.
Pineapple belongs to which family?
Bromelain.
Bromelase.
So that is why many enzymes are extracted from pineapple and also from papaya beta. Papaya is also a good source of enzyme extraction. So this is just an example to make you understand. Now we come to the third method. The third method was, see, give names in two words like you did binomial nomenclature, something like that only. In binomial nomenclature, you had the first word as the genus name and then the species name. This is how we had done. So isme bhi we are going to use two words, but first word will be the substrate and the second word will be the type of reaction that this enzyme is going to catalyze. Obviously, enzyme kuch na kuch reaction में help करेगा. So what type of a reaction it is doing? That will be the second word and the first word will be the name of the substrate. Let me give you this example. Pyruvate decarboxylase. This is the name of the enzyme. Pyruvate decarboxylase. So the first word is the name of the substrate. That means your substrate is what?
Pyruvate.
Pyruvate.
Your substrate is pyruvate and second type of reaction decarboxylase. That means this enzyme is involved. What is the function of this enzyme? Decarboxylation. Decarboxylation means what? Removing CO2. So it will remove CO2 from pyruvate. That is why it is known as pyruvate decarboxylase. First word से हमें ये पता चल गया on which substrate this enzyme is going to act. तो वो कौन सा substrate था बेटा? Pyruvate. So first word gives you the name of the substrate. And second word tells you the type of reaction. Because ये decarboxylation करेगा, मतलब ये pyruvate से CO2 को remove कर देगा. That is why the name of the enzyme is pyruvate decarboxylase. What is it? Decarboxylase.
Carboxylase.
हाँ जी बच्चों, these are the three methods that we have for naming. अब ये तीनों के तीनों outdated है. All three methods are outdated. We have a recent method, the latest method I'm telling you. अब यहाँ पे बहुत confusion होता था. I'll tell you why बहुत confusion होता था. Because when we talk about this, the root word में भी बहुत confusion था. In different countries, different people, root word was only different. We are calling it as malt. Some people were calling it something else. तो दो तीन तीन नाम हो जाते थे, but the enzyme was same only. For example, I called malt in India, and in some other country they call it halt. So rule तो क्या है कि root word के आगे ase लगा दो. So I am going to call that enzyme as maltase, but you are going to call that enzyme as haltase, isn't it, children?
Yes,
So there
ma'am.
was lot
Ma'am.
of confusion in this method. बहुत confusion हो गया because root word, the rule is very simple कि root word के साथ is लगा दो. But problem comes where? Problem comes in the root word only. Same substrate was known by different names in different places. Like I gave you an example of the substrate malt, maltose. We are calling it maltose. Maybe in some country they are calling it by some other name. तो rule तो वो यही follow करेंगे, but they are going to have a different name. And when it was checked for those 5 or 6 enzymes, it was found that we are talking about the same enzyme. But the root word was used in different languages as different word. So this system entirely outdated, never used, but वो common चल रहा है तो फिर भी use कर लेते हैं. Second system is very confusing. It is based on the basis of source. Can anybody tell me कि why this system must be outdated now? What क्या confusion हुआ होगा? First वाला तो I told you कि what must be the confusion. Can you guess बेटा इसमें क्या confusion हुआ होगा?
Ma'am, because one enzyme could be found in many sources.
Yes, beta, you've given the absolutely correct answer. Very good, excellent. Beta, one enzyme can be extracted from different sources. So let us not talk about families. Let us talk about general. Supposing there's an enzyme A. I'm getting it from papaya also. I'm getting it from apple also. Now there's a different family for papaya. There's a different family for apple. So again, wahi wala confusion ho gaya. So many enzymes, different names, but we were actually talking about the same enzymes. So there was a lot of confusion in the second method also. So this method was also discarded. Can you think about a drawback of the third system? Can anybody think about any drawback of the third system?
It might be too long.
One is no doubt it is too long. Second beta, if you see first word में तो वही वाला confusion आ रहा है जो first system में आया था. The first word is the name of what?
Substrate.
Substrate. And the substrate may be known by different names in different places again. And second word may be एक problem आता है. Can we guess out what is the problem which must be coming in the second word? It just tells you कि what function is the enzyme doing. यहाँ पे क्या confusion हो सकता है?
Because the reaction can be of many types, maybe.
Beta, one enzyme, excellent what she's thinking on. One enzyme can be involved in more than one reaction at a time. At the same time, it can act like a hydrolyzing agent. It can act like an oxidizing agent. It can act like a decarboxylating agent. You've certainly studied about redox reactions, where the
Yes,
same enzyme can help in reduction and oxidation both. तो फिर कैसे नाम देंगे हम लोग? There's a lot of confusion again. So there was, I mean, basically there were many confusions regarding all these systems of nomenclature that we had. Anyways, because they were followed, so we need to discuss them. Now I'm coming to the latest method of classification which is followed. This is a recent method. This is given by IUB. What does IUB stand for?
International Union of Biochemistry.
International Union of Biochemistry.
International Union of Biochemistry.
Yes, children, today I've taken up a very small topic. Hardly much is left in this. Because the chapter, this is the last part of the chapter. One more thing as a reminder, all 40 marks on Google form. Some MCQs, some one-word answers. So you have to be careful about writing the answers. This time it's an entirely objective paper. Just read your NCRT. Again and again, I'm telling you the same thing. All these things that we are doing, subjective part, that expression of answers, because last time what I felt is, sorry to say this, most of you were very honest, but in many subjects, what I expected, the answer sheets did not come the same. Some even, it was simply copy pasting better. So it is better we have an objective type of a paper where at least you'll read the chapters. That is very important. Even the letters in English, letters and articles were same for many students. I don't know, was it passed on from one student to the other student in a friend group, or was it copy pasted from the Google? Simply don't know. But in English also, beta, the article writing also, the articles are same, word to word same for many students. So there are two possibilities. Either you have a parallel group where things are being passed on, or it is being copied somewhere from the Google. Similarly, I felt in other subjects also. In class 12th, when I talk about biology papers, the same thing happened. Surprising things happened in class 12th when I checked the paper. There was one child who failed in class 11th, and of course, not naming the child, never. And the particular child failed in all 5 subjects. So obviously, the child was detained, not promoted to class 12th. But because of COVID, the government gave us instructions to promote everyone. Nobody would be detained. So that child was promoted to class 12th then. Okay. Now think about a child who failed in all 5 subjects, including the optional subject of physical education. The child fails in all 5 subjects, and then the child was promoted to class 12 because of COVID. Now, initial 1 month, the child did not attend the classes because according to the child, she was detained. After 1 month, we got an order from the CBSE that no, you need to promote everybody. So we promoted her, we informed her that now you are promoted to class 12, you can attend the classes. But she did not bother to come. So when we had these exams, your so-called half yearlies, this girl, she got 67 marks out of 70 in chemistry. You can discuss it, you can find out from Danish sir also. And the topper of our class, the highest marks were 64. So next day, what happens basically is when we have a class, we generally have an interactive session. Like I ask you questions, you give me answers. This is how we tried to do. But obviously, we did not get any of the answers. It is better I give you a paper where there is shortage of time. You need to do a lot of MCQs in a short span of time. Chali Bicha, you all are good children. Recent method. It was given by whom?
International Union of
National Union
Biochemistry.
of Biochemistry.
This is very simple. Enzymes are given some numbers, EC number. EC number. We call them as EC number. EC stands for enzyme commission numbers. What does EC stand for?
Enzyme commission
Enzyme
number.
Enzyme commission number.
This is very easy. EC1, oxidoreductase. Okay, see, both the functions, oxidation, reduction, both the work it is doing. Then this is EC1, oxidoreductase. Any enzyme over here first discovered EC1. Then transferases. EC2 were basically which enzymes? Transferases. EC3 were hydrolases. EC4 were lyases. EC5 is isomerases and EC6 is ligases. So we have given commission numbers to all of them on the basis of their functions. Okay. Now I can give you a few examples one by one. I'll give you a few one example of each. But abhi samajh lete hain. We have how many commission numbers?
6.
6.
6.
EC1 stands for
Oxide reductase.
oxidoreductase. Matlab both oxidation as well as reduction.
Reduction.
Like we had done electron transport chain. We had cytochrome oxidase. What did we have? Cytochrome oxidase. So if you remember about ETC, we had the role of this cytochrome oxidase. So this is acting both like an oxidoreductase enzyme, that is oxidation plus reduction. Let me talk about the second type of enzymes, that is transferases. Transferases, they are going to transfer something from one place to another. That is why such enzymes are put in the category of transferases. Let us understand this. This is glutamic acid. Okay. Glutamic acid, it is going to bind with pyruvate and alanine is formed. What is formed is alanine and alpha ketoglutamate. Now there is an enzyme over here that is glutamate transaminase. See, the name of the enzyme is very important. It is glutamate transaminase. Let's break this word. Glutamate means glutamate matlab glutonic acid se le liya. Transamino. It is going to transfer which group, children?
Amino group.
Amino group.
Amino group.
It is going to transfer this group from glutamic acid to pyruvate. And because of this transfer of the amino group from here to here, your compounds are going to be alanine and alpha ketoglutamate. So basically it is helping in transferring some group from one to the other one. Look at the name, glutamate transaminase. मतलब it is going to transfer from where? Transfer यहाँ से नहीं करेगा pyruvate से, यहाँ से कर रहा है. So हमने इसका नाम लिख दिया पहले. नाम से we can make a glutamate transaminase. From glutamate, it is going to transfer the amino group. So आपका क्या बन गया नाम? Glutamate transaminase. Which was the first category of enzyme? शुरू में लग रहा है बहुत मुश्किल है, but आप examples करोगे तो easy हो जाएगा. EC1. EC1 was which category?
Oxidoreductase.
Oxidoreductase. Again, I am telling you in your syllabus, बस इतना याद कर लो, EC6 sufficient. No need to do examples. Why I am taking examples is कि आपको अच्छे से समझ में आ जाए. That is it. So when I say oxidoreductase, what do you think is going to be the function of the enzyme?
Oxidation and occurs
Oxidation and
simultaneously.
reduction.
Oxidation and reduction.
So any enzyme which is performing the function of oxidation and reduction, we are going to put it in which commission number? What commission number will you give it?
EC1.
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 It is EC1. Second, EC2 is which enzyme?
Transferase.
Transferase.
Transferases. These are
Transferase.
major categories. Very good, beta. These are major categories, major groups. First one we have done oxidoreductase. Second is your transferase. From the name only you can make out transferase is going to transfer some some group from one substrate to the other substrate so that we get a new product. Just now we did an example where we had the substrate is what? Glutamic acid.
Glutamic
And then
acid.
yes, beta. And then pyruvate was there. The name of the enzyme was glutamate transaminase. So glutamate में से transfer करेगा amino group. So इसका नाम हो गया glutamate transaminase. Let's move on to the third one. Hydrolases. बहुत common पढ़ा है आपने. This is a hydrolyzing enzyme. This is a hydrolyzing enzyme. What does it actually mean, beta? बहुत जगह पढ़ा है हमने. This is an hydrolyzing enzyme. 10th में भी पढ़ा होगा. Hydrolysing enzymes or hydrolases are those enzymes which break a bond, but they break a bond by addition of water. So remember this category of enzymes, hydrolases, they will do what function? They will break bonds. What will hydrolases do?
Breaking
Breaking
bonds.
of bond.
Breaking
Breaking
of bonds.
How they will break bonds? By addition of what?
Water.
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 Let us do this one. This is a simple example. Starch. Starch in the presence of amylase. Okay. If we have amylase, the starch will break down into what? Maltose and isomaltose. Two products are there, maltose and isomaltose. आपको इतना डिटेल नहीं जाना है, बस इतना समझ लीजिए that starch में क्या हो रहा है. The bonds of starch are broken down. बेटा if you see यहाँ पे reactant सिर्फ one है. And how many products are there?
Two.
Two. That means यहाँ पे bonds break कर गए होंगे. That is why इसमें से एक reactant से दो चीजें बनी है. Very clear from this equation कि इस reactant we had just one, products we've got two. So bonds were broken over here. And this bonds must be broken by an enzyme which is going to break this bond by addition of what? Water. So water will be added over here. Only if water is added over here, then amylase can do this work. So amylase will break the bonds, but by what? By addition of water. Such kind of enzymes are known as?
Hydrolases.
Hydrolases.
Yes, beta, hydrolases. So which was the first category of enzymes? EC1. ऐसे करते हैं. EC1. What does EC stand for?
Oxidoreductase.
Enzyme commission number.
Enzyme commission.
Enzyme commission.
Enzyme commission 1. So EC1 enzyme category is known as which kind of enzymes?
Oxidoreductase.
Oxidoreductase.
Yes, that means what is the main function?
Oxidation and reduction reactions.
Very good. Now let's move on to EC2. EC2 is which enzymes?
Transferases.
Transferase.
Transferase.
So basically they are going to transfer something from somewhere. And the third category of enzymes, EC3, are which one?
Hydrolases.
Hydrolase.
They are very important enzymes, whether it is chemistry or it is biology. Hydrolases are very important enzymes. What is special about these enzymes, beta?
They break bonds
They
by
break
addition.
bonds
Bonds by the addition of water.
They will break bonds. So you may come across many enzymes where in chemistry also, whenever water is added, the bonds are broken down. So understand that the catalyst basically are hydrolyzing ones. Next, beta, lyases. They also break the bonds, but without the addition of water. So there is a difference between hydrolases and lyases. Hydrolases will also break bond. Lyases will also break bond. But if you look at this, lysis means breaking. The word lysis, if you remember, this lysis means what? Breaking down. And hydro means water. So lysis tab hoga jab water hoga. Then such enzymes will be known as hydrolases. But without water agar bonds break kar jaye, then no need to add the word hydro. Simply say lyases. So liases are those categories which will break bonds without the addition of water. If you remember in glycolysis, we had this fructose 1,6 biphosphate. There was an enzyme, aldolase. It was converting fructose 1,6 biphosphate to 3-PGAL and dihydroxyacetone phosphate, 3-phosphoglyceraldehyde. This was one step in glycolysis. अब इसको simple तरीके से समझते हैं. Fructose 1,6 biphosphate means 6 carbons. How many carbons in this entire substrate that I have over here? 6 carbons. Sorry. How many carbons?
6 carbons.
इधर एक ही reactant है, इधर दो products हैं. That means bonds to break हुए. And let's talk about PGAL. It has 3 carbons. Similarly, dihydroxyacetone phosphate also has 3 carbons. So 6 carbons here, 3 carbon plus 3 carbon, 6 carbons here. What is it very clear? That bonds have broken down. Between fructose, biphosphate, इसमें bonds are breaking, but water is not needed here. So these kind of enzyme, that is aldolase. देखो बेटा, amylase ने काम किया था, but it needed water. Aldolase can simply break the bonds without the presence of water. And this is your fourth category. I've written a wrong number. It should be number 4. So it is liases. This is EC what? EC?
4.
EC4. This is EC4. EC1 was?
Oxidoreductase.
EC2 was?
Oxidoreductase.
Transferases.
Yes. EC3?
Transferase.
Hydrolases.
Hydrolase.
EC4?
Lysis.
Lyase.
I hope everybody has understood the difference between EC3 and EC4.
Yes, sir.
Yes, ma'am.
Hydrolyzed and lysis. Let's move on to the fifth one. बस ये आज इतना ही करना है बेटा. That's it. EC6 तक. Nothing more we have. EC5 isomerases. They convert one isomer to another. They're simply converting one isomer to another. The name itself tells you isomerases. For example, glucose 6-phosphate. I'm talking about the substrate glucose 6-phosphate. And there's an enzyme, hexose isomerase. The name of the enzyme is hexose isomerase. If this enzyme is present, your glucose 6-phosphate will change into fructose 6-phosphate. अब ये हमने नाम क्यों दे दिया? There are 2 reasons. Hexose. Hexose इसलिए दे दिया बेटा, both of them are hexose sugars. Whether it is glucose or it is fructose, both of them are hexa sugars. So first word यहाँ से आ गया hexose. Isomerase, because glucose and fructose are isomers. So it has changed glucose to fructose. That means it has changed one isomer to the other isomer. That is why the name of EC5 is, what is the name of EC5?
Isomerase.
Isomerases. And the last, that is EC6, they're ligases. Ligases are joining enzymes. They help in the formation of new bonds. There were 2 enzymes which were breaking the bonds. What was the name of the enzymes?
Hydrolases and lysis.
Liases. अब हमारे पास एक enzyme ऐसा आ गया who's going to help in the making of bonds. They are known as liases. Pyruvic acid plus CO2. देखो बेटा. Okay. We are taking 2 reactants over here. Pyruvic acid plus CO2. Pyruvic acid is 3 carbon. 1 carbon comes from CO2. Both the substrates are going to join and we'll get oxaloacetic acid. आपको साफ दिख रहा है reactants 2 हैं, product 1 है. That means bond formation is taking place. Bond breaking नहीं हो रहा है. What is taking place over here is bond formation is taking. New bonds are being made. That is why 2 reactants converted into one product. The name of the enzyme is pyruvate carboxylase. You need not learn the name of these enzymes. Again, I'm telling you. 3 carbon in pyruvic acid, 1 carbon in CO2. Oxaloacetic acid is a 4 carbon compound. So बहुत clear दिख रहा है 3 carbon plus 1 carbon is giving you 4 carbons. That means new bonds were being made. So these are the 6 kind of enzymes categories. Now it is very simple. As soon as there is an enzyme, simply put it in a category. कोई फर्क नहीं पड़ता किस सोर्स से आया, क्या है क्या नहीं. Simply say EC1 enzyme, EC2 enzyme, EC3, 4. Though we have subcategories, of course we have subcategories also, but आपको इतना डिटेल में नहीं जाना है अभी. So I'll just be scrolling down very quickly for revisions. By the time I'll take your attendance, And then we'll have a question round on what we have done. Akash?
Present, ma'am.
Yes, beta. Just tomorrow we just have one class more. Aradhya?
Present, ma'am.
Okay. Anshika?
Present, ma'am.
Okay, dear. Ariba?
Present, ma'am.
Aryan Raj?
Yes, ma'am.
Okay, बच्चे. Ayush Patel?
Present, ma'am.
Okay. Then we have Drishti.
Present, ma'am.
Good. Mansi.
Present, ma'am.
Good, good. Shazir.
Present, ma'am.
शेजर बेटा आया करो, सिर्फ 2 दिन की अटेंडेंस हुई है इस मंथ में आपकी बच्चे. ट्राई टू बी रेगुलर. कोई प्रॉब्लम हो तो मेरे पर्सनल नंबर पे शेयर करना. देन वी हैव शेजर. देन वी हैव सृष्टि. सृष्टि,
Present, ma'am.
what happened yesterday? You were not there. You were so regular, but yesterday you were not there.
Ma'am, actually, I'm having cough and cold these days.
Oh God. Take care. चलिए, सोहम and Sony.
Present, ma'am.
Sony is there. Soham is also there. Very good. Uditi and Vaidehi.
Ma'am, please give Harshita entry.
Arey, sorry, she's out. Sorry, beta, I did not see. I was taking attendance. Uditi is here. Fine. Vaidehi is also there. Mahi.
Excuse me, ma'am.
Yes, dear.
Ma'am, please scroll down.
Ah, beta. Good, you reminded me. Keep revising. I'll be asking questions also. Good. I have done till Mahi. Then Harshita is already done. I hope I'm not missed out. All are there. The new boy has not come today also. Vivek Raj. He called me so many times after his admission. Ma'am, please add me, and now he's not there. Vivek Raj is not there, no? Ayush Gupta is absent. Let me see who are the absentees. Prabhav is absent. Raghav is absent. Ritul is absent. Sanvi. I don't know what has gone wrong with Sanvi these days. Shimail is absent. Pragya is absent. Ananya is absent. And Anshul Verma is absent. Vivek Raj is absent. So many children. I am anticipating what must be the attendance tomorrow. Tomorrow the festival. Chale bacha. Recent method very quickly revise, then I'll ask questions. Learn the categories. No need to learn the examples, but I'll show you the examples. They'll help you to learn them. That is the only reason why I've taken examples. See the examples also. That way you'll be able to learn in a easier manner. Two examples you can see from here only. Oxidoreductase, transferases, hydrolases, all three on your screen. Now, when you read about lyases, keep in mind the difference between hydrolases and lyases.
Ma'am, could you please scroll up to oxidoreductase once again?
Yes, Pichi. See, I'll just show you the first two, oxidoreductase, cytochrome oxidase. If you remember a bit, open your electron transport chain today. You'll immediately recall.
Thank you, ma'am.
God bless. Should I go down, children? You want to see the next ones very quickly?
Yes, ma'am.
And don't panic if you don't find this. Oh, this is not there in the book. This is not there. It's okay, even if it is not there. Most of you are targeting for NEET, so it's going to help you. Now the third and the fourth one. Should I show you the last category now, or you want to see this only more?
And last one.
Last one.
Isomerization. Yes, Vijay. Whenever you'll study with examples, things will always be easier for you.